Right, good afternoon, everyone. Welcome to the Hall of the Future here at Hub Week. We're here at Government Center for our Saturday session at Hub Week. So I am thrilled to be here with two leaders in the pharmaceutical industry. And I have Barbara Sosnowski, who I've just had a fantastic time talking to her and David. Barbara is the Vice President of External Science and Innovation Head for Pfizer's Cambridge Research Unit, and she oversees the identification of external opportunities for discovery to Phase 2B in several of Pfizer's research units. Next to Barbara is David Shields, and David is the head of Pfizer's Center for Therapeutic Innovation in New York, Boston, and Europe, and he drove up from New York to be here with us today. So thank you both so much for being here, and welcome to our Facebook Live audience. Welcome to Hub Week. So I want to thank you. I want to start off with Barbara, and we're going to talk about, as Zoe said, collaboration. But I want to first, why don't you set the stage for us and talk about how Pfizer really approaches R&D today?
Yeah, so we look at R&D looking at both internally as well as externally. So we know that there's great innovation outside of our four walls, and especially here in Massachusetts because of the intellect and the innovation that we see in Massachusetts. And what we try to do, what we want to do, is take our best capabilities that we have at Pfizer and marry those to external innovation that's brewing in either academia, biotechs, or other pharmas here within the area. And we combine that to make not just a 1 + 1 = 2, but to really make it a multiple of that. So we take the best of what we focus on in outside as well, and then combine that internally with what we do inside. And we have multiple ways of funding that technology or those assets, that collaboration. We have seed funding where we'd start new companies. We have an emerging science fund where we share part of the risk with our internal team, with a research unit, and we partner with an outside group. So they're really novel, creative ways, and these are in addition to the standard partnering like licensing and merger and acquisitions. So we really try to work with the scientists in the developing companies to say, what do you want? How is it going to be best fit for you as well as us?
Okay, fantastic. So you work a lot with scientists, but David, you work with scientists too in academia with your center. So tell us about that and why that center was created.
Thanks, Kathleen. So our Centers for Therapeutic Innovation are focused on how can we work together with scientists to jointly move forward their ideas, their concepts into a new medicine. So as Barbara said, we've got a significant R&D footprint internally. We invest, we have 25,000 researchers and clinicians that are moving the concepts, the ideas forward and advancing those into what's going to be our next medicines. But how can we identify all the new innovations that are coming out? We can't work in all spaces. We can't cover all the new ideas. So what we do is we try to identify external innovators, the smartest folks, guys who are pushing the boundary into new spaces that are not yet charted out. And can we work with them jointly? Not something where we'll fund, hand off and move on, but rather can we work hand in hand and try to bring those ideas forward, try to pressure test these new concepts, and then ultimately try to make them part of our R&D engine as we move forward.
Fantastic. So I want to push on that a little bit in talking about, you sort of both insinuated that there's new ways of operating. So I'm curious about those new ways of operating. Maybe Barbara, with you first. And then I also want to understand how you collaborate with each other and those two pieces. So you want to start there?
Sure. So when collaborating with CTI, Centers for Therapeutic Innovation, and David, first of all, we sit right next to each other in the Kendall Square. And so, we bump into each other quite a bit. So that's great, and it's, but when we're out scouting, we just don't scout for a particular area within Pfizer. We scout and try to fit, if it's a great asset or research collaboration, where does it best fit? Does it best fit within the research unit? Does it best fit with CTI? So we bring the science back, and what drives, it's got to be great science. And when it is great science, then we say, is it a CTI partnership? Is it a research collaboration within the research unit? Or should we form a new company? And so we need to ask those types of questions in order to find the best home for it.
All right, that's great. And then, so David, where do you come into play on all of that?
Yeah, so we're focusing on the ideas that are coming from the academics, from the clinicians, the folks that are at universities like the great institutions around here. How can we advance those ideas? But there's a change in how research is done in academia. It's no longer just research funded by the NIH, by foundations. There's a lot more relationships with companies. As Barbara said, there's new codes coming out. And so when we collectively go out and try to identify these new innovative ideas, we don't say what stage is it at, what might be the best funding mechanism. We just say, is it a good fit? Is this really an innovative idea? Is it going to be transformative? Can it be translated into a medicine? There's some great science that may not translate, but can we identify the science that will translate? And if so, then where does it fit in our organization? What does the investigator or the company want? There's many different models through which we can advance a concept. But so we work very closely with a partner to make sure from the outset we understand what their needs might be. Is it funding? Is it leveraging the resources from Pfizer? Is it working together to build up the concept? Maybe it's very nascent. Maybe it needs some more work, and then we can both contribute to that to see if we can get that over the edge.
Cool. I want to poke on something that both of you have said a couple times of great science. What's the definition of great science today? That might be a hard question, but I think that it's interesting to me. Is that sort of a lingo that you both have, or like how do you see that science has changed so much today? Like, what do you see as great science today?
Well, one thing we do stay within our core strategy areas. So oncology, rare diseases, inflammation immunology, internal medicine, and vaccines. So we scout in that kind of sandbox. And what we're looking for is not Me Too drugs. So these just can't be an add-on. We're looking for transformational therapies. Some of the example of that is immune tolerance for inflammation immunology. We're looking for cures. And so that's what we mean by, it has to be best in class, first in class, or only in class. And going into the scouting, you just don't take a look at the science. It's also got to be about, are we the fifth to market? Because we got to think about that. If we're fifth to market, we're going to, we won't be the best in class. And so all of those little parameters actually come into picture when we evaluate new opportunities.
Fantastic. And then you work a lot with academia. How do you see academia changing today?
It's changed so much in the last few years. See, our Centers for Therapeutic Innovation, CTI, has been going for 6-7 years now. When the concept was launched, we were seeking to address a gap in the market as we saw it. There's many PIs who had great ideas, they wanted to advance them, but they weren't necessarily ready to do all of the work that would truly pressure test those concepts. And we saw an opportunity to have a joint model where we wouldn't necessarily just take the concept, try to move it on ourselves, or just fund the academic and have them run with it themselves. Rather, we say, can we work on that together? At this stage, we're now at the point where we have 25, 26 programs in the portfolio, very robust, multiple programs in the clinic. But during those 6-7 years, we've seen a striking change in the academics as well in how they want to move the concepts forward. And so this is where the flexibility comes in. We may work with the academic at the front end in this joint co-discovery model, but then let's see how things evolve. As the concept matures, because it takes time, it's not going to be a 2-year runway going from the concept through to the drug. Does the academic partner still want to continue in the same vein? Do we want to continue in the same vein? Strategies, science evolves so quickly now. Can we adjust? So as the concept matures, maybe it is a great target, maybe it's a great medicine, the drug looks good, it looks like it could have legs, but should we maintain the same path internally? Do we stay with traditional Pfizer clinical development? Is this better suited in a new company? Is this better suited to a partnership with other companies that we might look to advance that? So we see a lot more flexibility from our academic partners, academics, clinicians, a lot more interest in exploring these different models. And we just need to be adaptable to what they want to do.
All right. So that's great. So it's a lot more different types of collaborations, a lot of different models. But a lot of it is about the incentives and the money. So maybe, Barbara, let's talk a little bit about that and sort of Pfizer's VC program.
Yeah, so Pfizer has Pfizer Ventures. It's led by Barbara Dalton, and it was recently revamped. And that announcement came out this year. They added $600 million over multiple years, but that $600 million is now added into the portfolio, of which there was already $500 million dedicated. So all in all, there's $1.1 billion in this in Pfizer Ventures. The other thing that was, I think, significant impact for Pfizer Ventures is they went from 4 people to 10. So they've added biology, they've added chemistry to help review new opportunities. And so they are looking to invest in early stage companies. They can be the lead, but they can also be follow-on and be in the syndicate for investing into newcos. But they've really taken us, and what I should say is that 25% of that 600 million is dedicated to neuroscience. So even though we exited neuroscience this year, we are still investing in early stage neuroscience opportunities because we still believe that there's value in going down that path.
Okay, very interesting. So we're in the Hall of the Future right now, actually surrounded by startups. And those of you on our Facebook Live channel and watching on video, imagine that there's all these startups around us, and you might be a startup too. So for those startups, how do they engage with you? How does an academic engage with you if they think that they have best-in-class science and they want to talk to you?
So, part of our group, we spend a lot of time at scientific meetings. We're all scientists by training. We spend a lot of time at posters, just reading San Diego. And, now that I've been at Pfizer for 8.5 years, I understand both sides of the coin, so to speak. And so I know what everyone's going through when they're going through that startup phase. So I'm happy to have people reach out and contact us. And we have regional scouts here in Boston, San Francisco, San Diego, all over the United States, New York. So it's and Europe, I should say, right? So.
No, awesome. So David, tell us a story. You've been working in this group for 8 years. Tell us a success story of like a technology or a something that you found, an academic that you found in a lab that you were working with, and something fun that happened.
We've got, I'd say, several different examples, even just in this area, and so perhaps we'll speak to some of those examples because those came out. They were completely nascent ideas from local labs. We worked with one PI in Boston University, had a small lab at the time, was just getting started, but had a fantastic concept, had some great genetics around an idea, wasn't even published, and in that instance reached out to us. As Barbara said, we have scouts who proactively go and find these ideas. At great events like this or at scientific conferences, we are there, but folks can go on to our Pfizer partnering pages or Pfizer CTI webpage and reach out directly, and we all, as Barbara said, work together. So if it's an idea we make sure it goes to the right home. In that particular instance, the PI came up with a scientific concept. We sat down, we saw this was something that needed some more work, so we worked on that together. We provided some funding, but then we're providing the resources as well. So it's not a sponsored research type of agreement. It's more this joint idea, collaborative research, where we can provide funding, we're providing guidance, but then we're doing work as well to ensure that the biology is advancing, and then we can start to bring in chemistry and so on. That concept has matured. It's gone all the way through to clinical trials at this time. The PI is still our partner in crime. We met with him this week for his updates. We provided ours, looking at the clinical development strategy. And that's perhaps where these partnerships are so important. It's not just saying let's work together for 1 year or 2 years, because again, that's not how long it takes to take the concept all the way through. So you can have the investigators' input at the earliest time when they had the idea for a new target, where it might work out. As we move along, how is it working? What's the mechanistic basis for this? And then as we move towards the clinical setting, what's the right indication? Where should we go? What is the disease? and then leveraging the investigator's expertise around the target, around the disease space, this can help us to better position where we place the medicine. And it's going to give it a better chance of succeeding because we're going to put it to the right patients. We're ensuring it's not going out to a nebulous population. It's going to be directed towards those that are probably going to benefit most. So we have several of those that have emerged from Boston. Like I said, the portfolio is robust at this point, and we've sourced some of those targets from all over the world. But a lot of exciting science.
That's really exciting. In that case, I sure some people in their minds, like, who owns the IP in that case? And perhaps it's nuanced and it's not cut and dry.
Yeah, so it's joint.
Oh,
Yeah.
so it's joint.
It's joint.
Okay, so that's really interesting.
Yeah, and the way we work is that we partner with the Institute to have an agreement in place with the Institute that covers everything from what might be the scope of the research, how many individuals might we fund, what's the timeline of funding, how does this work, what about publications? This is the bread and butter of the academic investigators, right? So they may bring funds in from us or from other sources, but at the end of the day, we need to ensure that great science gets out for the academic career, but also to advance the field as a whole and ensure that if this great discovery has been made by us jointly, that's been circulated, been socialized, and to bring the field up as a whole. The IP, what the terms are, all of that is written into our agreements.
Brilliant. And then on the innovation side and R&D side, Barbara, tell us the story.
Well, I think that, a good story is all about transthyretin cardiomyopathy. It's a disease that's caused by protein misfolding. It's a rare disease.
Okay.
And Jeff Kelly, in 1989, identified a couple molecules. He's out of Scripps Institute over in La Jolla. He identified a molecule that actually inhibited this protein misfolding. From that, he knew he took it far enough and he couldn't take it any farther by himself, so he formed a new company called FoldRx. That's based here, it was based here in Cambridge. And FoldRx developed it to lead molecules, and they took it to as far as they could take it. And then in October of 2010, Pfizer acquired FoldRx. And what it enabled us to do was to run very large clinical studies, 30 months in duration, with a large number of patients on people that had this transthyretin cardiomyopathy. So there's room for it for everybody because you have inception by the academic, you have proof of concept being made by the biotech, and then you have enabling it to get it to launched and out to be a product. And right now, this product is waiting to get filed to the FDA. But they couldn't have done it without having all three of those enablers.
Yeah.
And so it's a really great story about using academic, biotech, and pharma.
That's super interesting. So right here, we're in the Boston ecosystem. So that's a really thriving ecosystem with a lot of exciting things going on. But David, you just came up from New York. So talk to us a little bit about the difference between the New York ecosystem and the Boston ecosystem. I'm curious how you see that.
Different ecosystems. I think those are two examples of the places where we operate. As Barbara mentioned earlier, we also work across Europe. And there's very different, let's say, approaches to science, approaches with how the concepts can be advanced. And so it's one of the interesting challenges. How can we advance these different concepts best, being cognizant of what the investigators might be seeking, what the institutes might be seeking, and then also taking into account what that competitive landscape looks like. Boston is a fantastic scientific sandbox in which to operate because you have so many academic institutions, but then so many different companies as well. Large companies, numerous startups, as you say, robust biotechs as well, and we see lots of integration even across the companies. New York is somewhat more nascent, perhaps not quite as centered in and around the one area, but there's various operations that are coming up in New York, starting to have different biotech parks. We're based down the Lower East Side, where we have big companies such as Roche and Lilly and ourselves, many startups in there as well. And so now they're starting to see these biotech clusters come up in New York. Space is always at a premium down there, as you know, in Manhattan, so it's a different challenge. But again, great institutions down there, fantastic scientists, and ultimately that's what we're looking for. We don't hone in on a particular geography. There may be an individual investigator at an institute that's not within one of these traditional big clusters, if you will, but we follow the science and say, let's find a way to partner with those folks. as opposed to gravitate towards what might be seen as the bigger centers.
Oh, yeah, no, so you're
Yep.
downtown New York, and then, Barbara, go ahead.
Yeah, I was just going to say, that's why it's so important for us to be flexible.
Yeah.
And nothing's cookie cutter, right? It has to be, we have to go with how kind of the society and the civilization is in that hub. And, what works in Boston might not work in Europe, and vice versa. So again, we have to be creative, we have to be open, and we have to be talkative, like, what do you want? And how is it going to best fit both of our needs?
Right,
So,
right. Let's change topics a little bit from sort of the regional discussion to interesting new innovations and technologies that are emerging. So on the main stage on Wednesday, we were talking a lot about gene therapy. And so that's something that's getting a lot of hype these days. Barbara, you want to jump on that and talk about that?
Yeah, so we started our initial interest in gene therapy with a collaboration that we had at King's College. And that went really well, of London, and that went really well. And so we said, we wanted to initiate another collaboration with Spark for hemophilia B using an adenoviral-associated vector that encodes factor IX. And so we initiated a clinical trial with Spark, and the paper's been published, and it looks like potentially reduces the amount of exogenous factor IX that those patients would need to be taken to actually clot blood. From that, because that went so well, we went through, I would say, a build-to-buy kind of scenario where we spent simultaneously building our infrastructure on manufacturing and kind of the translational biology, and then bought Bamboo Therapeutics, which had a number of different assets in it. A significant one was for Duchenne's muscular dystrophy, which is in clinical trials right now. And then but they also brought a manufacturing facility to us. So now we're kind of poised to actually take those gene therapy vectors and not just run small clinical trials, but be prepared for launching them once they go through the development process. After acquiring Bamboo, we actually did an agreement with Sangamo on their hemophilia A program. And so, again, we're delivering factor VIII for that through an adenoviral-associated vector, and that's on its way. It's an earlier stage program, but we're really trying to build both our rare monogenic diseases into a larger platform where we can actually establish cures. I mean, that's the whole goal for this, is to develop cures.
To develop cures. So in hemophilia, for people that don't know, and correct me if I'm wrong, but I was in London talking to people about it too, that is actually the least rare disease, in that it has the most patients in it, and then it's people that have this for their whole life. And so you want to build on that. I think hemophilia is a really interesting area in rare disease that we could eradicate it potentially with gene therapy. And
the paper that was published, I know that we had dosed approximately 10 patients, and before they were enrolled into the trial, they had to apply exogenous factor IX to stop for clotting. And then after they were given one dose of the adenoviral vector encoding the factor IX, we've seen a steady level of expression of factor IX in their blood. And so they needed a minimal amount to actually clot. And at least over a year, we've been able to see that required a level of expression of factor IX. So it's been really great. We don't know how long it's going to go for. I mean, that's the question. But we are continuing to follow those patients, and we're looking to expand that into a larger clinical phase 2 trial.
That's awesome. David, Barbara said a word that I want to play with. See if you want to play with a little bit with me on the word platform. Platform is you can't go hit a rock anywhere and not hit a startup or someone in academia that wants to develop a platform. You want to talk a little bit about platforms? Have you seen any interesting platforms?
Yeah, the platform is one space that we're always going to look to. There's the emerging science around targets and concepts and new ideas, but everything's going to be driven forward by what that new platform is. And often we're seeing, not just the incremental, but more the transformative advances, or when a new platform is brought to bear. So if we're advancing a new medicine, we've got many different platforms internally. So if you think of the traditional terms, whether it's a small molecule, whether it's an antibody, whether it's a degrader technology, gene therapies now, many different means to advance that concept. That's what we can bring to bear and say, here's a platform. If you have an idea, we've got many different ways to bring that forward. We'll find a way to make this a success. We'll find a way to make this into a medicine. But can we move beyond that and say, how can we, whether it's identify new targets, whether we can advance approaches quicker? Had a recent collaboration set up with a company here, Crystal Pi. Right, again, this is leveraging a platform to more quickly do molecular assessments, to better enable structure. Again, this is leveraging great computational approaches, came out from folks from local institutions here. This was a startup. We advanced this advanced. We partnered with them, and now this is an example of a platform that's going to apply across all our different areas. It's not specific to a therapeutic area. It's not specific to particular biology or disease biology. This is going to enable us to much more quickly increase the processes. So those type of platforms is what we're looking for to get us much further ahead, not just have these incremental jumps.
All right, interesting. Barbara, you want to build on that a little bit?
So we also have been developing other collaborations. I know this was brought up yesterday during a live Facebook chat with Draper Labs. So Draper Laboratories is developing organoids, a multi-cellular organoid system that allows us to evaluate, they're human cells, but that allows us to evaluate our drugs on human cells in an organism, but not in an animal. And so we can evaluate what toxicity profiles, we can evaluate whether it changes the phenotype of these cells. I think that we're looking for ways to actually evaluate our drugs in a human setting without being in humans yet, because that you can't go there. But it's a way to evaluate the toxicities, the effectiveness of those. So Draper's been great. They're right around the corner from us, up at Kendall Square. And the collaboration has been very positive. That's a two-way street. We're doing things, they're doing things, but it's very interactive. We listen to them, they listen to us. It's kind of a win-win. It's all around good.
Sounds like a great marriage. Everyone's listening to each other. That's wonderful. So let's see. So let's talk about, we are here at Hub Week in the Hall of the Future. And David, when you and I were talking a little bit before, you were talking about you're really looking for not what are the trends now, but where are things going to be. So we're getting the signal where we're going to be wrapping up. So I want both of you to let's talk about, if we're here together in the Hall of the Future in 2025, where do you think things will be? What do you hope that we'll be talking about then? Where do you hope that you're taking the center in your work?
Yeah, this is precisely the space where we're playing. This is what makes it most challenging but most fun, right? Ultimately, we're trying to predict what's going to be the emerging science, what's going to be the science beyond that. We don't have those bedrocks, we don't have those platforms. And so this is what we're looking for in collaborators. So for the investigators that we seek, or for the folks who are coming in that we're looking to hire. Ultimately, what we're looking for are folks who are agile, who don't have the traditional boundaries, whether it's around diseases or platforms or modality or approach. We're looking for folks who can work across many different diseases, looking for folks who can call out mechanisms, call out ideas, call out concepts that will be almost genetic segment agnostic, disease agnostic. Can we find ways to repurpose, to combine different efforts, to leverage, to work across different fields, not just in the biology, but in the computational technology and beyond. So how best can we marry each of those to try and map out the space?
Yeah, I would hope that in 5 years, or maybe 6, that we could, for complex diseases like rheumatoid arthritis, that we could understand the different subclasses of patients that have RA so that we can treat them more effectively. In these complex diseases where you can't readily, right now, do precision medicine, it's you get one drug and it might be 100, let's just say 85% effective in 1 or 2% of the patients. It might be 50% effective in 10% of the patients. So understanding the differences between those, I think I want to go there next. I want to understand a full complexity of the patient. And so you need computing power, You need, for your drugs that are going to be produced, you need molecular simulations. It's, if you have precision medicine, you can, and we do on some of our oncology programs, but we don't have that benefit now, or if it's coming of age for more complex diseases. And so that's where I would hope that in the next 5 to 8 years that we'd be able to really tear apart a disease and understand it better.
Brilliant. I hope that we'll be there as well. And David and Barbara, thank you so much for this really interesting conversation. I think you gave us a great vision of the future and how you're working with Pfizer to take us there. So thank you so much.
Thank
Thank
you.
you.